Thank you, Joan. So as Joan mentioned, my name is Jordan Mora. I work here in the research department of the Waquoit Bay Reserve. Lots of familiar faces. We have some people in the crowd from some of the different watering monitoring projects I'm going to be talking about today, so that's great. And as Joan requested, oops, I always put this slide in so I remember to talk about myself. So these are a bunch of the different projects that I've worked on at the reserve during my time here. When I got started here, I was actually in salt marshes. I was helping set up our sentinel site down at the Sagelot Pond site and putting in permanent vegetation plots, elevation markers, and then since then have sort of shifted through a couple different projects and landed where I am now, doing both a little bit of salt marsh work, but mostly managing our water quality programs. And how I got started here, I grew up in Maine. My parents are still up there. I moved out to Ohio for undergrad and went to Denison University. Got my undergraduate degrees in music performance and environmental studies with a minor in bio. So my little fun fact is I also play the violin. And after that, I stayed on for Denison as their environmental program manager for a year. It was a little transition job they had offered at the university when I was there. So I got to sort of take a break from being a student, but not a full break from academia, which was a great way to take a little bit of a breather before I went and got my master's at the University of New Hampshire. And I was there for 3 years studying salt marsh ecology with Dave Burdick. And then after that, pretty quickly shifted over to Waquoit Bay. And that was largely because while I was in New Hampshire, I had a National Estuarine Research Reserve Graduate Research Fellowship, which they're starting to bring back, yay! They were no longer doing the fellowships for a number of years, but I think there looks like there's funding back in the budget to bring back the fellowship, so that's great. So I was a fellow with the Research Reserve System in New Hampshire, and when Waquoit was reaching out looking for someone to start in on their sentinel site work, I was in a pretty good place to make that transition. So that's how I ended up here. And I think that's kind of it. Did I leave anything out, Joan? Is that decent? So yeah, so we will get started. So I threw this outline in. I'm going to ask you guys to please leave your questions for the end, just because we do have a lot to try to get through tonight. There's a lot of information that I want to share with you, and so hopefully if you can leave your questions to the end, we'll actually make it out of here within an hour. But here's the outline. Give a little bit of an intro background to what I'm studying, what the different monitoring programs that we have here at the reserve, what we're involved in, and where the sites are, things like that, why we're doing it. And then also dive into some of the trends. I keep calling them trends and pulses because not everything we're going to look at tonight is actually a trend, something that is consistent and steady in its slope over time. So we're going to be looking at some of that, and then there are two main or interesting results that came out of my preliminary work that have to do with the nutrient data and then also our macroalgal data. And so we're going to look at both of that a little bit closer, and then just a couple take-home messages for you guys to share with your friends and family. So as an introduction, just some fun pictures. These are our 4 main water quality monitoring sites, and I'll talk about these some more, but some of these pictures might be familiar to you if you're from the area. So this is Childs River. This is the center of Waquoit Bay. This is actually Sagelot Pond in the wintertime. It's all iced over. And this is looking north, that's Washburn Spit, so right at the Minot Yacht Club. I'm not going to go over this too much because Joan gave you a little bit of an intro on this, but we are part of 29 National Estuarine Research Reserves. And of course, we are here on Cape Cod. And our state partner is the Department of Conservation and Recreation. And we also have a nonprofit friends group called the Waquoit Bay Reserve Foundation, and they've been great supporting our volunteer-based monitoring programs and a lot of other great education and outreach programs that we get involved with here at the reserve. So we're very grateful to them. So here's a picture of Waquoit Bay. This is actually the Salinity Gradient from the Massachusetts Estuaries Project that was finished up in 2012. And I like this graph, or this map, because of a couple things. One is I always forget to talk about what is an estuary at the beginning of my talks. So this map helps remind me that to talk about the estuary, and that estuaries are where fresh water meets the salt water. And I recently learned that estuary is actually from the Latin word aestus, which means tide. So it's literally where the tide meets the fresh water. And we have lots of interesting and fun habitats in our bay. We have an aquaculture, oyster aquaculture farm here on Sea Pit River, the Washburn Island oysters. We also have a lot of commercial cohogging that goes on in the bay. Some great fishing. We got bluefish, striped bass, lots of anadromous, well, a lot. We have 3, I think, different species of anadromous fish that make their way up into Quashnet River, and also I think a little bit over into Childs River as well throughout the season. We have 4 pretty decent sized salt marshes. And if you are familiar with salt marshes at all, you are aware that they are great habitats for migratory waterfowl and different bird species, in addition to lots of other organisms. So we have those wonderful habitats. And then we also have 3 remnant eelgrass beds still present in Waquoit Bay. So we have all of these great economic and aesthetic pleasers that the Waquoit Bay gives us. And so we have a lot of great resources here, lots of great significance to try to keep our bay healthy, try to keep it sustainable so that we can maintain all of these wonderful habitats that do so much for us. So a little bit of background on the eelgrass decline. I mentioned we have 3 remnant eelgrass beds, but back in the 1950s, the entire bay was covered in eelgrass. And over time, we have seen that eelgrass decline to pretty much gone for the Waquoit Bay proper. And we only see those 3 remnant beds in the sub-estuaries of the bay, which is really sad. We're going to talk about why that was. Some of the things that contributed to that, maybe a little bit of climate change, but most likely excess nutrient loading. So excess nutrients in our bay cause these algal blooms. This is a picture from right out here off the bluff in 2003. Really massive algal bloom that happened back then. Climate change, obviously we're getting warmer temperatures, and we're already seeing impacts of that in our water quality here in the bay. So already a really local problem for us. In addition to the sea level rise, but I'm not going to talk about that so much today because that's more of a salt marsh thing. More in my salt marsh talks. So why do we get these algal blooms when we have nutrient loading, and where does the nutrient loading come from? On Cape, our big problem is septic systems. So we have these septic systems in our houses, in our residential neighborhoods, and they leach a lot of nitrogen into the groundwater that ends up in the bay. And then we get these algal blooms here in the middle of the bay. That's just because there's so much more nutrients in the bay. We're getting a lot more algal productivity. And my little schematic, so if you have minimal nutrients and you have balanced algal growth, then you have lots of good sunlight that permeates down to your seagrasses like eelgrass. You have lots of oysters and fish and a happy little ecosystem. But if you have excessive nutrients from human inputs, for us, like I said, it's mostly septic inputs for Cape Cod. But if you go down to Chesapeake, their main issue is fertilizer, is agriculture. So both of those can be contributing factors. But what happens is we end up with an algal bloom, like that picture I showed you of the bay. And that decreases the sunlight that makes its way into the water column. The other big thing that happens is that algae at some point dies, whether it's the end of the season or just its natural cycle. And all of that biomass that's grown in the water ends up decomposing. And the major contributor to our eutrophication problem is that bacteria have to decompose all that extra organic matter. And if you've been to any of my talks before, which I know a number of you have, I like to try to drive this home, and that it's not just algal blooms. That's not really just the problem. When the algae dies and decomposes, the bacteria respirate. So they're actually taking oxygen from the water, using it to decompose all of that organic matter. And what ends up happening is it sucks all the oxygen out of the water column, and we end up with fish kills. We can end up with fish kills. A number of years ago, there was a pretty bad fish kill in Little Pond. So if you go away with anything today, remember that bacteria are probably part of the problem in eutrophication. So the mission for the reserve systems is to look at some of these different threats, these different drivers that are happening in our water quality, and try to get a handle on their trends over time. So we look at long-term data. This program for the reserve system was established in 1995. Our program here at the Waquoit Bay wasn't established until 2002. But we are taking lots and lots and lots of data and hopefully translating that into something that is useful for coastal zone management. So I'm just going to fly through a little bit of the process, what that looks like, all that data collection. One of the primary means of collecting data is we put out these automatic data loggers called sondes. And I meant to bring an example today, but I forgot. But this is me holding a 6600 series YSI data sonde at our Minot site. Those stay in pretty much year-round. This is actually the only site we can keep a sonde in year-round. The other ones have to come out because of the winter. But primarily they're in April through December. They collect 15-minute data on a whole suite of physical parameters. So temperature, dissolved oxygen, pH, turbidity, depth, salinity, and we also get a chlorophyll fluorescence data stream from these as well. So those are the data sondes. We also collect monthly nutrient samples at the same locations as those data sondes. So every month I go out with a pole and I put 2 little bottles in there, and I can take a grab sample of water. This is an ISCO sampler. It's an automatic water sampler. It collects data for a full 24 hours. It's a full tidal cycle. And then this is also a picture of our telemetry station at the Minot Yacht Club site. You can actually see all of our sonde data in real time from the Minot Yacht Club. So if you're interested in finding out what the temperature of the water is in the sound, you can go to our site and see it in real time. You can Google CDMO and look up our site that way, or you can actually get the link off of our website as well. So the other part of the monitoring that I'm going to be talking about today is our submersed aquatic vegetation and macroalgae sampling process and data. So we've done this survey 4 different times now. It was 2004, 2007, 2011, and 2016. 2004 and 2007 were before my time, and they had very strong people out in the boat taking these samples. When I came along in 2011, we had to incorporate the winch off the side of the boat because the ponar grab is just way too heavy for me to lift. So that's the only thing that changed there. So we take these grab samples with this ponar grab. It actually opens, and then as it hits the water, as you pull it up, it closes. So you get a nice grab sample of the bottom sediments and whatever macroalgae is sitting on the bottom there. Then we put it in a tray, sift out all the algae, put it in a bucket, rinse it over the side of the boat, spin it with a salad spinner, and then bag it and take it back to the lab to be sorted. And those get sorted by species and weighed dry. So we have a dry to wet ratio for those. I think that's all I want to talk about there. Oh, I did want to acknowledge that the Marine Biological Laboratory has been super helpful in collecting this data, especially in the 2016 sampling. They've been doing a lot of work looking at macroalgae in the Waquoit Bay as well. So they've been a huge support. Can't thank them enough. So these are the locations. This is a map of all the locations of sampling sites of data that I'm going to talk to you about today. We have the 4 swamp stations system-wide monitoring program. That is where the SONS and the monthly nutrient sampling is happening for that program. In addition to that, we also have a volunteer-based monitoring program called the Bay Watchers. And those sites sort of supplement and add to our suite of samples here. So we can incorporate both Eel River, Childs River, Quashnet, Hamlin, Jehu, and just get a couple more sampling sites in Waquoit Bay as well. So it really fills out the data set to have that volunteer program involved. And then the little yellow dots are all of the macroalgae samples that I analyzed. There are actually way more dots than this in some of the surveys that we've done, like twice as many at least. But these are the only points that I'm incorporating in this analysis because these are the ones with the longest data set. So now we're going to dive into some of the trends, some of the graphs and data that I've been seeing from our data. And these are some fun pictures that got my interest peaked in terms of whether or not these sorts of events are going to happen more often or less often for us as we go through, as we move forward. So you've seen this picture. This is the algal bloom in 2003. There was also a fish kill in 2014. That was in late July, early August when we get our lowest oxygen levels. And it was so rapid and so such high magnitude that we actually saw a shrimp kill. Lots of dead shrimp washed up onto the shore that summer. And then I think that beach erosion might be Jose. That's your picture, Adrian. I think it's Jose. Anyway, so here are some results from our 15-minute SON data. We're looking at temperature here. These are regressions over an annual mean. And a couple things I want to point out here. We have our 4 sites: Childs, Minot, Metoxet, Sagelet. I know that's hard to see in the back. We've got time on our x-axis here. So none of these regressions are technically significant yet. If you're looking at these p-values, you'll see 0.09, 0.07, and then Sagelet and Childs River are pretty far off. And what I would caution you on the significance values are, those are pretty strong trends, especially in the 0.07 to 0.09 realm. I would call those ecologically relevant. And I think it's important to acknowledge that waiting for a p-value to be significant you can be waiting too long, that we should really be acting sooner. Not that we can do much about our temperature problem, but again, just to keep in mind, these are ecologically relevant trends. And this is results, again, from our 15-minute data of dissolved oxygen. We're seeing declines in all of our sites. Again, Minot not showing something significant right now. I think that has a lot to do with the flushing that the sound adds to that site. But this is pretty extreme for us to acknowledge that over time we're losing, dissolved oxygen is going down over time. And again, more scenarios like the shrimp kill are a problem for the health of our bay and for the organisms that are living there.
Could you click back just for 1 minute, just so I can look at the numbers on the temperature?
Yeah.
Thanks.
Okay. All right, so now we're starting to dive into our nutrient data. And I know for folks that don't look at nitrogen data a lot, Some of this can be a little bit overwhelming, but we have dissolved organic nitrogen, particulate organic nitrogen, and dissolved inorganic nitrogen. I'm not going to talk much about what all goes into each of those different components of the nitrogen levels in our bay, but what I do want to make sure that you understand from this graph is that added together, so this is a stacked graph here, added together this makes total nitrogen. So this is our mean total nitrogen for that year. And we have year on the x-axis here. Total nitrogen is what our TMDLs, our total maximum daily load thresholds, are based on. So for us, trying to manage nitrogen in our bay, the top level here is what we care about, that additive result. So if you're looking at these trends, we can see maybe not too much of an increase over time, but 2010, 2011, we seem to get some sort of spike, pretty extreme peak in those years. So that's our first observation. The next observation you might notice is we have chlorophyll and Feo Feyten on that top graph. Those are pigments that tell us something about the phytoplankton growing and living in the bay. So chlorophyll is like a proxy for phytoplankton, and we are not seeing a very strong increase, or really any increase over the years for chlorophyll. That's not to say that Childs River is doing well by any means. If you'll notice, these scales are quite high. Childs River has very high nitrogen levels and very high chlorophylls. And that has, again, a lot to do with the problems that we see in Childs River with eutrophication. We'll talk more about that in a second. And then this is Sage Lot. So Sage Lot, if you guys are familiar with the area, is much less dense residential development around it. There's actually the sub-estuary there, or sub-watershed, is pretty much owned by the state. So we control a lot of that watershed, and which means we can prevent a lot of development and prevent a lot of added nutrients entering that part of the estuary. Which means if you're to compare these scales against the Childs River scales, they're much lower. So we don't see chlorophyll much of a 20 ever, really, but normally in the 10 range for chlorophylls at Sage Lot. And then our total nitrogen, again, much lower scale, and we still are seeing this odd spike in the 2010, 2011 range. So that's all I'm going to talk about for nutrients for a second. Now we're going to dive into the macroalgae and eelgrass survey. And this is data from all 4 years. So we've got each of our different sites here along the bottom and the 4 years within that site. And I know there's a lot of information in this graph, so I'm going to point out a couple of things that are of interest. First of all, the Zoster marina is our eelgrass that we get in Waquoit Bay. We do have it in here in this graph, so it does appear for those who are interested. And then the other interesting little side note about our estuary is that we are also starting to see new species that we didn't see in our earlier years. So Spiridea filamentosa is one of those species that we're starting to see more and more of in our estuary. I don't know there's necessarily anything wrong with that. It's just interesting that our species seem to be shifting. And that might have something to do with some of the climate change impacts we're seeing. Rupia marittima is also a species that started to get established in our estuary. And for those of you who may be familiar with this plant, it actually normally grows in a salt marsh. It likes to grow in the pools of a salt marsh. And what we're finding out is that now it's starting to grow actually in the ponds in the bay. So we're going to keep an eye on that because it might be starting to take over a little bit where the eelgrass used to be. Maybe not in a bad way, but certainly want to keep an eye on that. And then one of the other interesting pieces that I want to draw your attention to here is this is Sage Lot Pond. So historically very low nitrogen levels and low macroalgae biomass. But over time, we're seeing an increase in macroalgae. We still see our eelgrass, it's still there, but we're starting to see a lot more macroalgae species get established and sort of proliferate in there. So that was an interesting finding. But I think that that is a pretty well understood phenomenon because, again, we have a lot of nutrients in our bay. So likely some small incremental increases in our nitrogen that's getting into sage lot may be explaining the increases in macroalgae that we're seeing there. However, the decreases that we're seeing in Childs River are a little bit more perplexing. This isn't something that, you know, historically we've seen in our estuary. Lots of macroalgae, yeah, we got that. But declines in our macroalgae, that's a little curious. So anyway, we have a 12-year spectrum here that we're looking at, and over that time, we definitely are seeing some declines in macroalgae. We aren't necessarily seeing the same thing in our nitrogen levels. We have a little bit of wiggle room in there, some high years and some low years, but in general, pretty steady. So, you know, that piqued my interest. Why would we be seeing decreases in our macroalgae in Childs River? You'll notice it also happens in Eel River too. I didn't highlight it because it's not significant, but that's a pretty strong trend as well. These samples that we take have a huge variability. We can get 0 grams in one grab and then right next door get 2,000. So, I mean, we're talking maybe 100 feet in difference. So really high variability. It's really hard to get statistical significance in these analyses. So who's driving? Why are we seeing the pulses that we are in the 2010 range in our nutrient data, and why the decline in our Childs River data? And these are pictures of maybe some of the things that could contribute to that. Residential development, storms, runoff from our tributaries. So the first question that I always ask myself is it real? As a technician, as someone who collects a lot of data, I am very skeptical of data always, because I know about all the things that can go wrong with it. So I looked into some other databases to try to see if I could corroborate that those same peaks in those years. And it turns out our Waquoit Bay Watcher data does seem to have some elevated nutrient data during those years. However, the Waquoit Bay Watcher data, those sampling events weren't necessarily on an outgoing tide. So I think that trend got subdued a little bit because they were actually collecting on an incoming high tide. So the water got mixed a little bit, and that trend's a little bit less strong in that data set. However, if you look at the Buzzards Bay Coalition Bay Watchers data, which you can look at online if you're interested, they also are seeing, you can look at the 2010 through 2013 range, and they also see pretty high nitrogen values. So that made me feel like, okay, well, at least I'm not crazy and our data isn't totally wonky. And it made me speculate that probably if we're seeing something on a regional scale like that, where Buzzards Bay is also seeing similar trends, that we might be looking at a climate change, or at least climate drivers inputting or impacting these data. So I looked at temperature quickly at our sites. This is our 15-minute data again. We do see metoxit and sage lot show some pretty high temperatures in 2010, but not so much in Childs River and Minot. So this might explain some of it, but doesn't look like it explains everything. So then I looked at our discharge data. We have a USGS water gauge in Quashnet River right down the street. And so I was able to look at our discharge levels over the year. And this is actually pretty interesting. So this graph, although it's kind of crappy, I really like it. Quality-wise, it's a little less than ideal. But the graphs that they provide on their site give you the median daily statistic for what's calculated over a 28-year time span. So this little orange line is what the Waquoit Bay, we'll say, is used to getting. It's kind of the pulse that it's sort of adapted to. But in the years where we're seeing really high nitrogen levels, we had really high discharge rates that were much above that median value. And if you line up those discharge measurements against our nitrogen, they seem to line up. 2010 was a big year, and then we see some decreases more recently. That's not seen so well in the dissolved organic nitrogen values, but certainly in our dissolved inorganic we do see that sort of come down again. So there were a couple of big storms that impacted this as well. Some of you might be familiar or remember the storms that we had in March of 2010. I remember these storms very well, and I apologize for everybody who's already heard this story. But I was living in an apartment complex in an old mill on a river. And when this storm hit, I had to leave my apartment because it was going to get flooded. So I remember this storm very well. There was a lot of snow melt and a lot of rain. Rhode Island flooded in areas. So that was a really big storm, and it happened in March. So a couple of these different peaks seem to correlate with late spring or early spring, late winter sorts of events. So I looked at the monthly discharge rates against, this is actually the log of dissolved organic nitrogen, but I looked at all of our different nutrients, and we do see that March and April end up being the biggest deciding factors for our summer nitrogen levels in our estuary, which I thought was pretty interesting. As another piece to this puzzle, we are seeing increases in our precipitation and in our discharge values over the last few years. So this USGS station, so that's the one on Quashnet River down the street. And then this, the precipitation data is from a DCR, Department of Conservation and Recreation station in Falmouth. So all really close local data that we can sort of harvest from our area, which is really exciting and great that we have all these long-term data sets that go back to, the late 1980s. So the slopes of these lines are very similar. They're clearly very much intertwined in terms of their relationship, but also worth noting that, again, all of these impacts, we might see more of them down the road. So now we're going to go back to our macroalgae question in Childs River. Again, I'm going to ask myself, is it real? And there are a couple of papers that have been written looking at respiration and photosynthesis in the water column in the benthos that have been published in the neighborhood, one for this bay, actually for Childs River work, and also work that's been done at Narragansett Bay. So there seemed to be some corroborating evidence that something along these lines could be happening, where we're seeing less photosynthesis, less productivity in the bay or in these waterways. This is just a picture of Sarah Foster and Wally Fulweiler's crew taking those samples in Childs River, because we actually helped with some of that sampling. So this is a graph from their paper that was published in 2014. Again, sorry for the not ideal quality here, but what I want to acknowledge is that they're looking at rates of respiration and of daytime productivity. So oxygen getting added to the water column. And these rates look at the difference between the current being 2011, 2012, compared to historic rates from the early 1990s. And in pretty much all scenarios, except for some winter months in the productivity, the historic rates are higher than the current ones. So that indicates that there's been a slowdown in the metabolism, our bacterial metabolism, and then also our primary productivity. And here are some of the theories that they provide in their paper as to why this might be. sulfide concentration buildup. That could happen from anaerobic respiration. And environmental factors, and some of the ones they list are increased temperature, decreased wind speed, and changes in the timing of phytoplankton blooms. But I was mostly curious about this decreased wind speed piece. And in their paper, they suggest that summer wind speeds, based on our our wind data that we're collecting at the carriage house, that our summer wind speed data has that is reduced in speed over time. That they think they've found some significant results there. So I wanted to dive into that a little bit more. This is a wind rose of our data from the carriage house for July and August. This is just to point out that we do have primarily southwest winds in the summertime. And southwest winds would create a lot of mixing for most of our estuary, but in particular Childs River, shuffling some more water up into that part of the estuary. And we've done some work with a researcher at the Jacques Cousteau Reserve, who's also a professor at Rutgers, and he's run a couple analyses of our dissolved oxygen data and our dissolved oxygen data from Childs River and also our wind data from the carriage house. And we think we're seeing about a 2-hour lag, just means that from the wind peak wind speed, a 2-hour lag to when we think we're seeing the highest correlation to high dissolved oxygen levels. So we get some strong wind, that moves the water around, causes some more mixing, and increases dissolved oxygen in Childs River. So if we put all these pieces together, we know that Foster and Fulweiler found a significant drop in summer wind speeds based on their paper. We also know that wind seems to be important for dissolved oxygen in Childs River, that we do see a correlation there. And that if we're seeing less wind, then we are probably also seeing more hypoxic events from reduced oxygen levels, and that might be shifting benthic microbes towards anaerobic respiration. And that can increase sulfide concentrations in your sediments. And that is not fun for anything living in those sediments. It's toxic for most plant species, and it's a very stressful environment for any sort of macrofauna. It can also, and I'm sure Sarah will talk about this more in her talk in a couple of weeks, can be leading to a reduction in our denitrification happening in that site. But I'll let her talk about that when she gets here in a couple of weeks. Oops. So combined, I suspect that in Childs River, the reason that we're getting reduced macroalgae is a combined effect of eutrophication and climate change that's driving down productivity. So here are your take-homes for the day, for the evening. The first one is that heavy spring rain and discharge are clearly playing a big role in our nutrient levels in our bay. We aren't necessarily seeing increases in those over time, but we do seem to see these pulses from really high rain events. And one of the reasons that we might not be seeing steady increases in nitrogen, even though we know certainly you can drive down the street and see a whole bunch of new development going in around the bay. I know I've looked at our Google Earth images and seen quite a bit of development just in the last 5 years. But one of the reasons that that might not be showing itself, reflecting itself in nitrogen levels in the bay is because in the same time scale, we have also been experiencing decreasing atmospheric nitrogen due to improved air quality policies that are keeping refineries and things from spewing lots of pollution that ends up as acid rain and hitting the estuary that way. So we might be seeing a little bit of a balance between some improved, good EPA regulated policies, and that's balancing out any of that residential build out in our estuary. Or at least that's what Valiella's team is saying. They published a paper on that just, I guess 2 years ago now. And then the decline in the macroalgae in Childs River is likely a combined effect of historic long-term eutrophication and also climate change. So that reduction in wind speed, the increased temperature, all of that can lead to lower dissolved oxygen levels. So the important thing to recognize here is that as we move forward with coastal management, we really need to be taking climate change factors into account as we consider our nitrogen removal. So as I was mentioning, that total nitrogen level that all of our total maximum daily load thresholds are based on, that's a threshold that's been built out and is on the records for the town to try to meet for our nitrogen loads. But if we're getting increases in that from things like more precipitation, higher temperatures, reduced wind speeds, all of that is going to play into how well those thresholds actually keep up and can actually provide the healthy estuary and water quality that we need in order for all of those great habitats to thrive. So going forward, and hopefully Webner will participate in those negotiations and those talks moving forward about what's our best plan of attack to try to remove nitrogen from the estuary so that we can meet our thresholds and make sure that we can improve the health of our bay. This is a long list of relevant and cited literature that I used for this presentation. I think this presentation will be available online on our website. So if you're interested in any of these, you can check them out on the site there. A few acknowledgements. I had so many interns and volunteers that I should have listed on this slide, but there were so many of them they wouldn't fit. So lots of interns and volunteers have helped collect all this data over the years. We couldn't do it without them. Rick York works for the town of Mashpee. He's also been super, super helpful with all of our macroalgae data collection. Ivan Valia's lab, obviously with the macroalgae and eelgrass monitoring as well, has been a great support for us. Those are some of their funding resources for the last round of work that they've done. The Waquoit Bay Reserve Foundation, I want to make a call out to them for their help with the Baywatcher program, our volunteer-based monitoring program. And also our former and current Webner staff. This is a lot of data. It spans a really long time. There are people who have worked here, worked on these projects, and have now either retired or moved on to other things. So I want to acknowledge them as well. And with that, I'll take any questions.